Okay. All right. I don't know how long this is gonna take. I'm hoping it doesn't take too long, but you never know. So we'll see. But yeah. Okay. So lecture 15, just a couple of things that I wanted to talk about. Is these so like the different going from the AMP like the MP variant to the DP variant. There's individual kinases, right? So adenylate kinase, GMP kinase, and then UMP CMP kinase. That's the main thing I want to talk about there. And then for the broad specificity kinase, all of the DPs get phosphorylated by NDK. It doesn't matter what the DP is; it gets phosphorylated by NDK.
Oh, yes.
Period. Right. So the from AMP to ADP or from GMP to GDP specific kinases. So adenylate kinase for AMP to ADP, for GMP to GDP, GMP kinase, right? And then for UMP CMP kinase, that would be for UMP CMP to UDP CDP, right? Yep. And then for the DKs, any of the DKs does not matter. Up till the triphosphates, it's NDK. And here's some cool little pathway stuff if you want to look at it. And then cytodylate synthetase, this just comes from UTP. So for UMP to this, you'd have the UMP kinase. From UDP to UTP, you'd have NDK, and then from there you can just make CTP and UCDP. Cool. And NDK is reversible, by the way. NDK goes both directions.
Oh,
So NDK can go to the triphosphate version or back to the diphosphate version. Uh, Ribonucleotides are the RNTPs, not the DNTPs, the normal ones. So like ATP, for example, is that a DNTP or is that a RNTP? It's an RNTP. So right. So and then DATP would be the D version of that. So just remember to try and remember those, otherwise you will forget and everything will fall apart and we'll all be sad. So for Yeah, so the number of RNA building blocks is higher. The reason for that is just because we use it a lot. So like cell cycle, all that stuff. Cyclic AMP, if you remember from like Bio 200 and stuff, that kind of thing. Second messenger cascades. There's also just a lot more RNA in your cells, and like DNA is like kind of set in stone once it's been used. So and then cofactors as well, like NADH, FADH2. Those are all adenosine derivatives. So you need a lot of normal nucleotides. And I'm not going to go over this. You guys know the difference between these two. OH, H. I sure hope that we've been here long enough that we know the difference between these two. um, so yeah, and then let's see. Critical to keep the four DNTPs in balance. Yes, that makes sense. You need to replicate with all four of them. Deficiency of DNTP is lethal. You can't replicate if you don't have DNTPs. So, and then excess of them can be mutagenic. Just means that it can lead to more mutations. So. Yeah. And then here is an example of like what happens. So once we get to the DP stage for any of these, we do R to make the DNDP form, and then we use NDK to phosphorylate again. So just remember that what goes into R is the diphosphate version, not the triphosphate version. The diphosphate goes in, and then the diphosphate comes out, and you phosphorylate it. You got a triphosphate.
is it the same ending?
Yes, RNDP. Yes, NDK literally just takes any diphosphate, turns into triphosphate. It doesn't matter. Yeah, when you say RNDP, you mean like this?
Yeah.
Oh yeah, RNDP just the N just stands for anything. That's all it means. Yeah, the N just stands for AD whatever, right? Yeah.
Yeah, but R and D, right? They're both like the opposite of the right.
Oh yeah, so the riboform and the deoxyriboform.
Phosphor to triphosphate side.
Yes. Yep. Yeah, this guy does not care. Anything gets phosphorylated by him when it's got two phosphates on it. Yeah, you can imagine that what that means is that the binding site is only specific to two phosphate groups. It doesn't care about the base. For this, for the R stuff, just know that NADPH goes in, NADP plus comes out, and water also comes out. Hopefully the water coming out makes sense. It's kind of like if this is not the action mechanism, it's kind of like you're protonating the OH group and it leaves the leaving group almost, and then the H goes in there instead. And this is The thyroid-auxin pathway. I don't think it's that important to know all this. You can study it on your own time if you'd like, and I will talk about it in a video eventually, but I have not posted that because I didn't think it was going to be on your test. So, because it said up until lecture 14, and then they changed it. So, yeah. So here's the enzyme. This guy is a big boy. He's very large. 250 kilodaltons is massive. It's an octamer. You've got 2 alpha subunits, 2 beta subunits. And let's see here. It involves a free radical mechanism. Generally speaking, whenever you see an OH being replaced with an H, it's a free radical mechanism. Generally speaking. And it's also highly regulated. So, here's the structure of it. I don't think this is that important. I think it's there for you. I don't think it's that big of a deal. What you should know about eventually is the regulatory sites and all that stuff, but we'll talk about that in a second. You should know that it's catalyzed by a tyrosine radical, but other than that, I wouldn't be too concerned about it. Here's the mechanism. Hopefully it makes sense if you've looked at it. Again, they're not going to test you what the mechanism is. So, I mean, maybe they'll test you on the mechanism. I don't think they will. That would be kind of demonic if they did. I don't think they will do that, though. Yeah. The whole at least tells you what he's not going to test you on. So, okay, so now we talk about how do we balance things, and I'm not gonna look at this slide. I'm just gonna go straight to this slide. Right. So we have the activity site, the specificity site, and the catalytic site. The activity site decides if it's on or off. Right. If I bind ATP, it turns on. If I bind DATP, it turns off. That's it. Right? That's all I want to say for this. So that's what the activity site literally decides: is the enzyme turned on or is it turned off? If ATP binds, I turn the enzyme on. If DATP binds, I turn the enzyme off. That is it. Specificity is this is the site that decides what the catalytic site ends up binding to. So if I bind DATP, for example, I will bind CDP and UDP in my binding site. If I bind DTTP, and this is made, by the way, from the later pathways from this, then I'll bind GDP and make DGDP. And then I'll do NDK, and then I'll bind DGTP after making that, and then I'll make ADP and DADP. And then once this is phosphorylated, I make DATP, and that turns the whole thing off. It's like a chain, if you want to think of it like that, right? It's like the product feeds back into the enzyme, and it changes the thing. So, let's see here. Yeah, and what you should know is that in order for any of these bindings to take place, like DATP, GGTP, all that stuff, you need to have ATP bound into the activity site. The activity site is turned on by ATP. There's 3 different sites. Yes, that is what I'm trying to get at here. Yes. There's the activity site. Is the enzyme on or off? There's a specificity site. The specificity site is like, oh, which nucleotide do I want to bind to this site? So if I bind DATP, I'm going to bind CDP and UDP. If I bind DTTP, I'll bind GDP. If I bind DGTP, I'll bind ADP. And then if I bind DATP, then I'll turn the whole thing off. Here's the oligomerization states. Oligomerization states. Jeez. Tongue twister. Yeah, so I mean, this is here if you want to take a look at it. I think if I'm not mistaken, this is the inactive form. So the octamer is the inactive form. Basically, you take 2 of the enzymes and you slap them together. You're basically taking 2 RNRs, because one RNR is A2B2, right, or alpha-2 beta-2. When you take 2 of them and combine them, you get alpha-4 beta-4, and that's the inactive form. But here's the active form. You have the alpha-2 beta-2 tetramer, and then you have the weaker affinity form here, which is just alpha-2 beta-2, and they're just separate from each other. These 2 have to come together to form the alpha-2 beta-2 complex, which is the active form. So you can imagine that like this tetramer forms when you bind ATP. Yeah, essentially, if you want to think of it like that. And it says here, like, it associates to form this complex when the alpha subunit, when all 3 of them are occupied, by the way. Sorry, not just the ATP. All three of them have to be occupied. Activity site has to be occupied, selectivity site has to be occupied, and the catalytic site has to be occupied. All of it has to be occupied. All three of them. So when you occupy all three sites on here, I don't know, whatever, something like that, then you'll get to make this, and then you can keep going. Genuinely, note card. This, I do not think they're going to ask this. I highly doubt it. It is there. The rest of this, I do not think is that important. I'm not going to lie. This is important. These reactions, I hope you kind of write these down on your note card. So there's two things I'd write on my note card for sure. This pathway and this regulation. Yeah, unless you have it memorized. If you have it memorized, then don't worry about it. But I'm going to make the assumption that we don't have it memorized. So copy down this on your note card, and then copy down this pathway on your note card.
Is this regulation?
Yes, this is the regulation I was talking about. That was like I am very very concerned. Yes, yeah. But yeah, I mean the one. This is hopefully this is like relatively self-explanatory. I know it's a lot of information, but um, this is just saying like how do we make TTP from like UMP and stuff like that and CDP. So CDP and UDP we use RNR to make it into the D versions, right? Then we use NDK to phosphorylate. And then remember CTP, the only difference between CTP or cytosine and uridine is that the top carbonyl group on uridine is replaced with an NH group, right? So if I just remove the NH group from cytidine, I get uridine. And so that's why they're saying use a deaminase.
Do you think these?
Yes, if you want to think about it like that. Yes. So yes, you have a deaminase, you get a DUTP, and then for whatever reason, nature decided to just remove all of them. So we remove both phosphates from the UTP and turn it into DUMP. And then we use thymidylate synthase to make DTMP, thymidylate kinase, and then NDK again. The one thing I want to keep in mind is what is the difference between UMP and TMP? There's one difference. What is the difference? Methyl group. Where does the methyl group come from? THF. It comes from 5,10 tetrahydrofolate. Please remember that. It's an important cofactor. THF is used in a bunch of other things. You'll eventually do it with amino acid stuff. It'll come back. And that's what they talk about here. This, yes. N5, N10 methylene tetrahydrofolate. So that is what donates the CH3 group to the UMP to make the TMP. So also would write this down in your note card if you feel like you're going to forget this. But yeah. And yeah, here's just an overview of it. Honestly, if you wanted like one slide to like just copy on your note card for all the nucleotide stuff, this is the slide. This is the slide I would just copy down because it has everything in it. But yeah, it doesn't encompass the regulation very well, right? So it doesn't tell you like what things like what comes out of this step and all that stuff, right? But yeah, but yeah, let's actually get into the exam now because that's why I think most of us are here. So I assume most of you have probably done this test already. So I won't like beat around the bush here and all that stuff, but okay. So yeah, I realized that I left my phone. Let me go grab my phone because I need to make sure I don't lie to you. So grab the answer key.
Exam study, exam 2, lecture 4. Sure. Does two transfer this or all real transfer?
You guys are being gaslit by your own prof.
Yeah, it's a exam 2. Yeah.
What? See, oh god, dude.
Teacher, right?
You did not teach, yeah, whole top 15s. Oh, did he? When does he ever know? Bless him. I'm sure he's a wonderful man, but yeah, outside of the classroom, I'm sure. Okay, let's go ahead and start cooking here then, gang. Okay, so which of the following statements best describes the properties of RNA polymerases? Okay, so RNA polymerases. I'm thinking transcription. I'm thinking they produce RNA in the 5' to 3' direction, stuff like that. Right? I'm just like trying to get my mind jogging here. Like, what am I thinking of when I see RNA polymerase? And I'm thinking of what kind of things does it use? Does it use dNTPs or does it use normal NTPs? Right? Stuff like that. So for this, we're just going to go through each answer, eliminate if it sounds dumb, and then go just based off of that. So RNA polymerase requires a primer to initiate transcription similar to DNA polymerase. No, you do not need a primer for that. So get it out of here. Catalyzes the polymerization of ribonucleotides in the 3' to 5' direction. Nope, should be in the 5' to 3' direction. Right? It reads 3' to 5'. It produces 5' to 3'. So delete there. The sigma factor in prokaryotic RNA polymerase is essential for elongation, but not for initiation. So this is actually backwards. Sigma in prokaryotes is important for initiation, not so much for elongation. So that's reversed. And the rest of it's actually true. Eukaryotic RNA polymerase 2 does require the mediator complex for proper transcriptional act. That is true. It's the first part that is wrong. So RNA polymerase has intrinsic helicase activity that unwinds the DNA duplex during transcription initiation, but its efficiency is modulated by nucleosome positioning in eukaryotic cells. Okay, I'm gonna be honest with you, that's a mouthful. Honestly, I could see that being right, so I'm just gonna go to the next answer and see if it's a better answer, and then if it's a better answer, I'll just pick that one. So eukaryotic RNA polymerase 2 requires transcription factors. Yeah, I know dang well that's true. It requires general transcription factors and a mediator, okay, for protein promoter recognition and initiation, where a prokaryotic just relies on sigma for binding, and that's it. I think E is a better answer than D. D, I think, is like, I think D is okay, but I believe, if I'm not mistaken, there actually is, you do need actual helicase when you do transcription as well. You can't just rely on the intrinsic activity. Because remember, if you try and do transcription and you just try and push the RNA polymerase, you get this really weird positive supercoiling that ends up tightening the whole thing. That was something that was discussed in your lecture a little bit. But hopefully the reason why E is a better answer makes sense, though. It just makes more sense, yeah.
Would it be the chromosomes for the what like the thing that comes in there? Like you just said.
Oh, you're right. I think it would be it might be topoisomerase for the unwinding. Yes, it'd be topoisomerase. So that's actually another reason that this would be wrong in that case.
Well, then wait, well, did because I thought it not like it was like something about it's more on the side of like, oh, Arctic is a 35 you right and it's 17 is open at once, but
Yes.
opens and closes all the way down. So it says you doesn't.
Yeah. I'm just trying to. I don't think it has intrinsic helicase activity. That's why I'm like,
So I thought RNA it did open up itself. Like you don't need to heal it these things.
yes, that part is true.
So it's a weird wording.
It's just weird wording. Yeah, I think the big thing is like unwinds to me sounds like the topoisomerase thing you were talking about. I think you're right that it does have like somewhat intrinsic helicase activity because it is able to split, but it doesn't unwind it. That's why you need a topo in there to actually unwind the supercoiling.
The front.
Yeah, the front, exactly. Yeah, because as you push towards it, right, it like pulls and tightens it. So you do need to unwind that. But that's not helicase's job. Helicase's job is to chop it.
Oh.
So, yeah, that's another reason why this would be wrong, actually. That's actually a good point. So, yeah, hopefully that makes sense, though. I'm like trying to rush through this a little bit. Okay, which of the following RNA types is least likely to be involved in direct translation but plays a role in regulatory gene expression? Okay, so anything involved in translation, so rRNAs, mRNAs, tRNAs, those are all in translation, right? So any of those are just wrong. They're not answers to this question, right? Because it says least likely. So in that case, mRNAs, they're obviously involved. In translation, rRNAs are obviously involved in translation. tRNAs are obviously involved in translation. Now we're just between microRNAs and snRNAs, small nuclear RNAs. Gonna be honest with you guys, I believe these, right, snRNAs, these are more common in like ribozymes, right? Or SNPs, right? Yeah, like the spliceosomes. Sorry, yeah, not ribozymes. Spliceosomes. Yeah, thank you. So these are more involved in spliceosomes, right? So I mean, technically that is involved in translation somewhat. I mean, I guess, right? Because you have to maybe do some RNA processing. Maybe you do use a spliceosome in that process, right? So, yeah, so I can see that being uh, possible there. But microRNAs for sure play a huge role in gene expression. You guys haven't talked about microRNAs too much in 441. In 442, you'll have a whole section on them. Um, but microRNAs, basically what they are is like they are small pieces of RNA, and they will bind to RNAs and stop their translation and stuff like that. It's like post transcription, if you will. Post transcription, you can have a microRNA that binds to something else, and that causes the you to stop doing any sort of translation on that RNA. More important in 442, but since it came up here, just keep that in mind. Hopefully, the other three make sense. So these obviously are directly involved in translation. SNRNAs. Uh, I think the big thing with snRNAs is like they play a regulatory role in gene expression. They don't really play a regulatory role. You know what I mean? They play a role in like after you make the transcript, you edit it, but they don't regulate it. If that makes sense, they just help make the protein or make the RNA. So yeah, hopefully that makes sense.
For like what protein expresses as the alternative splicing or just splices them?
Yeah, that's fair. My only argument to that would be that when we talk about regulatory, we're talking about like, oh, is it upregulating expression or downregulating expression? Not is it making a different protein. But I mean, it's a valid point. Like, I mean, like, I think that's what they wanted you to get at when they talk about gene expression. Is it more expressed or less expressed? That kind of thing. And microRNAs, they literally stop you from expressing things. Like they will bind to RNAs and stop translation. So that. Okay, for 3, which of the following best describes a distinguishing feature of eukaryotic RNA processing that significantly impacts transcript, export, stability, and translation? Okay, so I'm looking at things that have to do with, um, especially with this, like stability and translation. I'm looking for 5' cap poly A tail. Those are like some of the most important things in eukaryotes, and that's literally how we stabilize all of our mRNAs. Otherwise, they would just explode. So anything to do with 5' cap. So actually, if I just look real quick, I can actually eliminate, because some of these just don't have anything to do with 5' cap or that, but let's just read through them anyway. So 5' cap facilitates nuclear export. That's true. It does help with translation initiation. This is also very true. And protection from exonuclease degradation. Also true, because it's got that weird reverse cap. That makes it's like the guanosine is like reversely attached, or if you remember right, it's like weird. It's like a 5' to 5' meeting, right? So that helps it with exonuclease degradation prevention. The poly A tail of eukaryotic mRNA promotes nuclear export and stabilizes the transcript, but is not required for translation. This is wrong. If you remember that one slide that I talked about where it turns into a U shape, it is required for translation initiation. There's one slide exactly. It's a slide that has like the Kozak sequence and stuff. That slide shows you that translation initiation requires this U shape for eukaryotes, and so it is required for initiation. That is wrong. Right, but that's like a one slide thing.
Usually, like, so the poly is how like she kind of interacting.
It's the poly A tail. There's some middle ground proteins. So there's like the polyadenylate binding proteins, and then there's some other extra proteins that connected to the 5' cap. I can't remember which lecture that's on. I think it's on translation 1, if I'm not mistaken. Go up. I think there should be a slide that like talks about like the like Kozak sequence, like 5' stuff like that. I think it was translation 1. It was relatively early on. I think. I think it was translation 1. I could be wrong. Is it transcription?
It's transcription.
Maybe it's transcription.
For the U?
I think there's translation. It was translation. No. It was translation. Yeah, I thought it was translation 1.
But yeah, I mean, for boarding and stuff. I should have.
Yeah. Hey, let me know if you find it, but yeah.
Oh, yeah, that's question 13.
It is, it's 13, okay, so I messed it up, yeah.
Yeah.
Oh, so it is translation 1 then. Okay, okay, cool. Yeah, okay. Anyways, we'll keep going. Eukaryotic mRNAs undergo splicing to remove introns, which help regulate stability and translation potential. Okay, doesn't really impact transcript export, I guess is my thing here, right? This is true. It does undergo splicing to remove introns, but like that doesn't really affect transcript export. It just helps with creating more diversity in our proteins. So I'm not the biggest fan of that answer. Alternative splicing in eukaryotic mRNAs can modulate transcript stability and influence translation efficiency. This is the same thing that I said above. Like it's not really um it's not really that important for like transcript export and stuff, you know. This is true for like, yeah, I mean, sure, it can help with stability maybe a little bit, and maybe it'll help with translation efficiency, but it doesn't really have to do that much with transcript export, right? It has more to do with diversity of proteins. So whenever you see splicing, right, that more has to do with creating different isoforms of a protein so you can get diverse protein structures. So I would say D is not the best answer here. And then for E, RNA editing in eukaryotic alters nucleotide sequences, impacting transcript stability and translation. Gonna be honest with you guys, didn't really talk about RNA editing. You don't really edit RNAs that often. I mean, like maybe you can do a single nucleotide polymorphism where you adjust one of them, but that's not really a good answer for why to the best describe a distinguishing feature of eukaryotic RNA processing, right? If I want a distinguishing feature, I mean, theoretically, this could surely be done in prokaryotes. Like, who's to say it can't be done in prokaryotes, right? Who's to say that I can't alter the mRNA of a prokaryote, right? So that's not the best answer. When I'm looking at these kinds of questions, especially when it's saying best feature of a eukaryote, I'm specifically looking for the 5' cap and poly A tail because those are very unique to eukaryotes, right? Prokaryotes don't have a 5' cap and a poly A tail. That's a very important thing to keep in mind. So, what's the other thing like that prokaryotes don't have? That's like really important in this translation kind of thing. Yeah, they don't have a nucleus, right? Which is why it doesn't. I don't have to do this whole nonsense with the poly A tail G 5' cap, right? There's no nucleus. So it like as soon as it's transcribed, it gets snapped onto a ribosome, right? As opposed to in eukaryotes, right? I have to go to the rough endoplasmic reticulum and all that jazz. So yeah, A is the better answer here. Okay, which of the following statements best distinguishes the mechanism of a self-splicing introns from spliceosome-mediated splicing in eukaryotic RNA processing? I feel like these questions are so long. So I'm looking for anything that has to do with like using the spliceosome, right? The spliceosome protein complex with the snRNA. That's what I'm looking for. So self-splicing requires ATP. No. Never was there ever a mention of ATP being required to do splicing, especially self-splicing. So no. Group 2 self-splicing introns utilize ribozymes to mediate their excision without protein factors. Okay, yeah, that's technically true if I'm not mistaken. Whereas spliceosomal introns require dynamic ribonucleoprotein complexes. That is for sure true. Right? I definitely like this answer a bit more than A. Spliceosomal splicing occurs exclusively in the mitochondria. No. It occurs. No, it does not occur exclusively in the mitochondria. It occurs everywhere. Right? Self-splicing introns are recognized and removed by snRNA. Nope, that's not. This is for spliceosomes. Right? snRNAs are on spliceosomes, so they are not for self-splicing introns. So that's wrong. The presence of a specific nucleotide sequence within introns that facilitates lariat formation and exon joining is essential for self-splicing introns, but not for spliceosome-mediated splicing, as they do not rely on intronic sequences for splicing. This part is for sure wrong. They definitely rely on intronic sequences for splicing, right? That part isn't correct, so I would say this is wrong as well. So B is definitely your best answer there. Yeah. Which of the following best describes HIV reverse transcriptase in viral replication? For this, I'm looking for something that says, oh, we take our RNA, reverse transcribe it to DNA, something like that. Reverse transcriptase. I just realized. Yeah, okay. Reverse transcriptase synthesizes viral RNA directly from host genomic DNA. No, right? Reverse transcriptase reverse transcribes. It goes from RNA to DNA. It does not go from DNA to RNA, right? I think there is a subunit of it that is of DNA polymerase, but that's not what's going on here. So HIV reverse transcriptase functions independently of other viral proteins and host-derived tRNA as a primer during replication. I wouldn't say it functions independently. I mean, like host-derived tRNA. I'm gonna come back to that one. That one's a bit weird, right? So I mean, maybe, but like a function independently is a bit weird to me. Like I mean, like you do need the other viral proteins to get the thing to actually integrate properly, right? You need an integrase, you need a polymerase, you need all this stuff. So maybe, but I'm gonna come back to that. So unlike cellular DNA polymerase, requires a host-derived primer to initiate DNA synthesis, but does not require a template. This is for sure wrong. Requires a host-derived primer to initiate DNA synthesis. It does require a template, right? You can't just synthesize out of thin air, right? You gotta use that. But no, you don't know who threw that.
No, I was actually.
Oh, you're falling asleep. I see. Yeah. But yeah. I think the big thing here is like does not require a template. I mean, it does require a template. Because it would require the viral RNA as a template, right, to actually make the DNA. So it does require a template. So HIV reverse transcriptase is a monomeric and no, it's a dimer. It's a dimer. It's got an RNase as well, right? It's got an RNase and it's got a reverse transcriptase. So it's not a monomer. Exclusively catalyzes DNA polymerization without adding additional enzyme catalyst. No. Also, no. What do you mean it only catalyzes DNA polymerization? It's literally reverse transcriptase, right? It has to reverse transcribe. It doesn't do DNA polymerization. So, functions as a dimer. Okay, now we're talking. Okay, because we bought a dimer possessing both RNA-dependent DNA polymerase. Okay, and RNase H activity. Holy Christmas come early. E is the best answer here. So reverse transcriptase functions independently of other proteins. I think E is just a better answer, right? B, I feel like maybe you could argue for, but like also it doesn't really act independently, right? Yeah, that also is a bit weird. Independently of other and host-derived tRNA as a primer during replication. That is also a bit strange. The tRNA. Yeah, yeah. Yeah, I'm pretty sure the tRNA is not host-derived, if I'm not mistaken, but he said it's host-derived? Okay, yeah. So even if it is host-derived, though, I think the big thing for me is like saying it functions independently of other viral proteins is a bit strange. Yeah, it would require a tRNA. Also, like, it does need the assistance of other proteins to get, yeah, like the RNase and stuff, and all that. I think that's what I was trying to get at. Either way, E just better describes the question's answer. You know what I mean? Like what the question specifically asking for, E describes that better. Right, where E is like everything about E is correct, whereas with B I'm a little bit like I don't know. So that's like one piece of advice I would give is like if you're between two answers, right, and one of them is just like you know for sure it's like everything about it is good, and the other one you're like iffy on, stick with your gut. He does do that a lot. He does a lot of this thing where he like gives you two answers that seem very correct, but like you know one of them is right for sure, and the other one like seems good, but like it's almost like he's trying to trick you because he's giving you extra things that you didn't learn maybe, and you're like, oh, but maybe I forgot to study that. So he does do this a lot. Yeah, so I would say like stick with your gut. If you see an answer and you're like, "Oh, I know everything in this is correct," probably the right answer. So yeah, I'd go with E on this one. Second genetic code in protein translation. Okay, this is literally tRNAs binding to their codons, plus like the addition of the amino acid to every tRNA, that kind of thing, right? That is the second code. So refers to the ability of amino acyl tRNA synthetases to recognize and correctly charge tRNAs with their cognate amino acid. Yes. If I didn't want to, I don't even have to read the rest of them. That is literally what I was just talking about, right? The second genetic code is, does the tRNA bind to the codon, first of all? And second of all, does the tRNA get charged with the correct amino acid? That is it, right? The second genetic code refers to the redundancy of the genetic code that allows multiple codons to no, that is not the second genetic code. Right. The second genetic code describes the ability of the ribosomes to proofread. No, not really. That's just proofreading. That's not the second genetic code. Second genetic code is a mechanism by which mRNA modifications directly influence ribosome selection. Nope. Don't even talk about that when it comes to genetic code. Genetic code, we're talking more about like the matching of certain things. Second genetic code is the alternative decoding system used by mitochondria. No, he's obsessed with mitochondria. He's putting mitochondria answers on this test. I'm not going to pick any of the mitochondria answers. There's no mitochondria involved in this, okay? So, I apologize if I'm going quite fast as well. I just realized that like I took way more time than I wanted to on the review for lecture 15. So, yeah. Hopefully it's not too fast. But you can always rewatch if you need to. Okay. Who here is able to read these diagrams? These. Are you able to understand what's going on in these?
Yeah, that is the introns, yeah.
Okay, cool.
And a lot.
And you can recognize where the poly A tail is in the 5' cap.
Obviously. Yeah.
Yes, exactly.
That's Gino.
Yes.
That's either 2 or 4 is the function.
Yes. So yes, yes, that's a great way of thinking about it. Yes, 2 or 4 has to be the 5' cap or poly A tail. So let's talk about this, right? So which position has the reverse 5' to 5'? Fancy way of saying which one has the 5' cap. Okay, yeah. I remember seeing this when I first did this exam. I was like, what are you talking about? But yeah, 5' cap. So the way that we look at this is, right, we have to figure out which one is the DNA and which one's the mRNA. The way you figure that out is which one has the big loops, right? That is the DNA, like Hayden was saying, right? That is the DNA. That means that this top one is the DNA, this top one here, and the bottom one is the RNA. Okay? And so now all we have to do is we have to look and see where it's complementary matching. So if you see here, like you see how it's like connected almost in those middle areas, that's because it's binding complementarily. Right? The DNA and the RNA are binding together, basically. Because these are both single-stranded, by the way. And so when you go through this, you're going to look and you're going to see, okay, eventually I'm going to get to this point here where like this section here is like really wacky. But like what happens here is like this, if you zoomed in enough, this actually goes down like this and becomes here. Right? The red is what I'm drawing there, right? So it does this. That is the mRNA. Right? Okay, so first of all, we can actually do this one pretty easily. How many introns does it contain? Just count the number of loops. 1, 2, 3, 4, 5, 6. Circle the letter. There you go. 6 introns. Good. Okay. For this, the way that you tell is one of the ends of the mRNA is going to have a poly A tail, which is not going to match up with anything in the host genome. And if you look, 5 is the genomic DNA, remember. 4, though, right, has this little section coming off of it, poly A tail, right? Everything else matches pretty well. So 2 is going to be the answer to this, because 2 is going to be the opposite end of that. So yeah, there's really no way to tell, based off of this, which side is the 5' cap without using the poly A tail as a guide, like a guidance thing, if that makes sense. So we know that this bottom strand is the mRNA, right? Because of the fact that the top one has the introns in it, right? And so because we see this extra like bit coming off, that must be the poly A tail. Because we know the bottom strand is the mRNA. So if that's the poly A tail, the other side has to be the 5' cap, just by process of elimination.
Why would you call it phosphate linkage, not like one C?
Because it is a triphosphate linkage. There's two phosphates. It's like the 5' nucleotide of the RNA does this weird attack thing where you remove the two phosphates off the end of it, and then you attack it to a GTP molecule, or I think it might be a DGDP molecule. I can't remember exactly off the top of my head. But you attack it to a GTP molecule, and you only remove one of the phosphates. There's like a 3-phosphate linkage in the middle. This would be on lecture 11? Transcriptional processing, I think. I think it'd be lecture 11. Um, double check that for me. Yeah. Okay. Is lecture 11? I like how I've got these on lock right now. I've got the lectures locked down.
See the 6 slide. This is
Whatever Hayden's got there. Yeah. It is a triple phosphate linkage though, right? Yeah, like cross the top. Yeah, okay.
it's like okay. 1 5 gets methyl.
It also gets the methylation.
Okay.
The 7 methylation is there as well.
So what the linkage? Okay.
The linkage is a 5' to 5' end, yeah. So yeah. Okay. For 8, right? Identify the correct statement regarding so here's one thing I want to point out. You do not know what this is. Guess what? We didn't know either when we took this test. We didn't know what SAM was when we took this exam. We had no idea. But doesn't matter. So here's the thing. So you can eliminate pretty easily on this. So in cells, we'll start with 1. It's important to generate enzyme cofactors such as coenzyme A. I know that's true. NADH, yes, I know that's true. And FAD. If you don't NADH literally stands for I think it's nicotinamide adenosine something, and then FAD is flavodeno something, right? Dinucleotide, right? So that's how you know. When it says adeno in it, adenosine. So these are for sure made by that. And I mean, even if you didn't know what this is, very conveniently there is adenosyl in the name. So I think you can infer, even if you didn't know, right, that it uses adenosine. So 1 for sure is going to be correct. So anything that doesn't have 1 in it is going to go bye-bye. So those are gone. In cells, the vast majority of ribonucleotides are converted into dNTPs to support DNA synthesis and repair. Don't know about that one, guys. Remember that you have way more normal NTPs than dNTPs. You only ever need dNTPs at one point in the cell's life, really, for replication. Anything outside of that, heck no, right? Remember, your body's main source of energy is ATP. That alone probably outcompetes all of the amount of dNTPs you have. So I would say that this is probably not true. They can be used to generate cyclic AMP and signaling molecules. Well, I'm going to make the assumption that it's true, just because cyclic AMP has to come from AMP, right? Which is going to come from a nucleotide, an NTP, or an NMP in this case. And ribonucleotides can serve as a substrate in other metabolic reactions to generate activated intermediates such as UDP glucose. So this is a little bit of a flashback to 440, but I mean the answer is yes, because you've literally seen it do UDP glucose, right? So it can be used to generate activated intermediates. I'm trying to think of other examples. I mean, even hexokinase, for example, right? It uses ATP, right, to activate glucose. So that kind of thing. So it is used to activate intermediates. So I would say based off of this, 1, 3, and 4 are your best answers. So that's what I would go with. So D. Okay. Examine compounds 1 and 2 below. These 3 points. Please do not get these wrong. Leon, I'm looking at you. I'm looking at you. Okay.
600.
Okay, so compound 1 is a pyrimidine. Well, I don't know about you, but that is not a pyrimidine. What is that? Yeah, what's the actual nucleotide? Guanine, yes, thank you. It is guanine. What is this? Uracil, yeah, it's uracil. Okay, and Is this a DNTP or not?
R.
It's not, right? Because it's got a double OH. And then this is obviously a DNTP in that case. If you were to name these, what would you name them?
GT.
I mean,
So GTP. Okay, well I don't know why it's black. So I would agree with GTP on this one. What about this one? Yeah, DUDP. Yeah. So that. Yep. And actually, that just gives you the answer. Actually, so I'm not even gonna bother going through the rest of them. Compound one is that and this. Yep. So there you go. So yeah, that's an easy way of doing these. You can usually figure out what the question's asking by looking at what this is giving you. Whenever you see these kinds of questions, it's usually asking what's the identity of it. So you just use that. Yeah, you got it in your last exam, right? I mean, could he could theoretically? I don't. I find it hard to believe that he would. But he could. You never know. Okay, now we do R and R. The molecule below is shown to is bound to ribonucleotide reductase. Okay, the specificity site. Okay, not the catalytic site, not the activity site. Specificity site. The specificity site decides what binds to the catalytic site. So yeah, what is the next immediate step of the DNTP synthesis? Okay, so what is this? DATP.
Oh, it should have.
It turns off, right? Or that's what you thought it should happen. Yeah, it's bound to the specificity site, not the activity site. Yes. Let me put. Do I have that slide still? Hold on. This is probably where it's helpful to pull up that slide. Yeah, this is where I'm telling you just write the table down. I'm trying to remember if I had it memorized. I think I might have. I think at some point I had it memorized. What, this?
No.
Yeah. So here, let's just find it. So we're binding DATP here. So it'd be CUDP and CDP and UDP. Yeah. That's why I'm just saying just write it down. So B and C. Yeah. If it said activity site though, yeah. So they were trying to trick you on that. I actually remember this question, and I was like, "Ooh, you sneaky guy, trying to screw me over here." So yeah. uh, oh, you guys didn't do orotate, huh?
Yeah, that's dumb as well.
Well, you guys didn't talk about it, did you?
No.
You guys didn't talk about orotate, did you?
I feel like what inhibits cat.
Yeah.
I don't. Oh, did I miss that lecture? Which lecture was that? It was in 15. Where did I miss that? What?
It's in two ways. Like you reviewed it in lecture, like as you went back to lecture, geez. Yeah, maybe went.
He mentioned orotate in here? Really? I must have just missed that.
It's like two. Like you went through it all in that lecture.
What do you mean it's in lecture 2?
Because you were being
Oh my. Wait, so it was. When was it? It was.
in lecture 2.
Yeah.
You brought so in class we talked about it. What's that this slide?
You're kidding me. You're saying lecture 2 from like.
Yeah. Why?
Oh man, bruh. Okay. Well, he expects you to know it, sounds like.
Yeah.
Yeah.
Oh, did he? Did he talk about it in lecture 2?
He went back from lecture 2, and that's why the day was to lecture 16. He also did lecture 15 stuff because things got pushed down. It was like,
Oh dear.
constantly questions.
Yeah. I think I have a feeling I remember what's going on here. Okay. Yeah. I guess like for this, yeah, it kind of is just a case of do you know it or don't know it. Yeah, so you observe that the level of metabolic intermediate orotate is decreasing in the cell. What type of feedback inhibition is occurring? Just I think the best thing to remember here is that like orotate is structurally similar to UDP and UTP if I remember correctly. It's very similar. I'm trying to remember the exact structure, but it's very similar. It's literally the same except you just remove this, right? Literally, what happens? If I'm remembering this right, I think with CAD you are literally when you take the PRPP, right? You literally decarboxylate this thing off, and it's not, yeah. So I can't. I think like CAD is for the formation of this, though, if I'm not mistaken, right? But then there's like a decarboxylation step after, I think, right? Yeah. So there's like a decarboxylation step plus the PRPP, and so you end up with just this thing, right, attached onto the PRPP, which is just uridine. It's just uridine. So that's why I'm saying like this is the best answer here. Yeah, I don't think he. Yeah, I think there might be a slide, and if we have time at the end of this, I'll go back and try and find the slide. Um, I'm sure it's in there. We'll come back to this though, just to be sure. But yeah. What are the most abundant and least abundant? Come on, gang. Surely we know this, right? Um, so most abundant for sure is ATP, right? You literally use it for energy. I sure hope it's the most abundant one. So we can eliminate anything that says this. The least abundant one I think get maybe a little bit tricky. But you should no. You should never have DUTP, right? It is formed in the pathway to make DTCP, but it only lasts for a split second. Because if you had DUTP, guess what, gang? Have fun with DNA replication. You're going to start putting U's into your DNA. So DUTP is not very common at all. Right, so that's just something to keep in mind. You might have to memorize that, but hopefully that makes sense. You like never see DUTP. And if you go back to the pathways as well, like I don't even think DUTP is a thing. I think it like goes down to yeah, I think. Oh, I'm sorry. No, it does go down. It does start with DUTP, but immediately goes down to DUMP, right? And then it goes into DTMP, and then you keep going. But, yeah. You're studying the proofreading mechanism. Okay, cool. By introducing mutations in the editing domain. Okay, editing domain. Which of the following observations are made? Yeah, I remember this question very, very vaguely. This is a bit of an annoying one, if I'm not mistaken. Yeah. So, so mutant thing leads to mistranslation and misfolded protein. Okay, cool. Lowering valine concentration reduces misacylation, but some misincorporation still occurs. An active site mutation restores accurate amino acylation despite missing editing domain, but alters isoleucine binding affinity. Structural analysis shows the editing domain interacts allosterically with the active site. So I think the main ones that I really care about here are these top two. So like a mutant isoleucine RS lacking the editing domain, so no editing, right? Misacylates tRNA this with valine. So that means that I'm supposed to have isoleucine, right? But the editing domain is screwed. So I import valine in there instead, and it still is fine. And it leads to mistranslation, misfolded protein. Great. Okay, that's fine. Lowering the valine concentration reduces the misacylation. Okay, great. So it's no longer acylating to valine that much. But there is still some misincorporation. That's important to know. And then an active site mutation restores accurate amino acylation despite missing the editing domain. So if you mutate the active site so it specifically binds to the isoleucine, works fine, or yeah, isoleucine, but alters the affinity for it. And then the structural analysis shows that the editing domain interacts allosterically with the active site. Sure. Basically, I'm looking for anything. What for? Yeah, it's just saying that like the editing domain of amino acyl tRNA synthetase interacts with the I guess like the active site in that case would be like I want to say like the binding of the tRNA itself, if that makes sense. It's like the binding of the tRNA itself to the thing is a separate process from the editing domain, right? I think that's what they're talking about when they're talking about that. But so which one's the best conclusion? I'm just looking for anything that talks about how the editing domain like stops it from misacylating. Because that's what the first 2 points we're talking about, right? The first 2 points are basically saying that when I break the editing domain, I'm screwed, right? Like I misacylate and I get these weird translation things, right? That's what the top two are saying. That's the most important thing. Does that make sense? That's the most important thing from this question. So the editing domain is unnecessary. Bro, what are you yapping about? Yeah, it's very necessary, right? For accurate amino acylation. Yeah, for sure, it's necessary, right? We just saw up here, like point one literally says that it gets messed up. So amino acylternary synthase relies only on ribose. No, nope, nope. It definitely relies on its own proofreading, right? So it does its own proofreading. That's the important part. Editing domain prevents misacylation, but active site mutations can restore accuracy with altered kinetics. That's kind of literally what the third point is saying. It literally says that it restores accurate amino acylation, but alters the binding affinity. So the third point lines up pretty well with this. It also lines up pretty well with what we talked about in 1 and 2, right? How it prevents misacylation. Because when you break it, it starts misacylating. So hopefully that makes sense. And then misacylation with valence is irrelevant. Bro. No. So misacylation with valence is irrelevant to protein function. That's not true. It is relevant. It also doesn't help explain the findings. These findings don't have anything to do with that, really. These findings are more talking about the editing site. So, um, reducing valene levels eliminates the need for proofreading. Showing misacylation depends only on substrate abundance. I mean, I guess, but I wouldn't say it eliminates the need for proofreading. That's a bit extreme. You know what I mean? You still need proofreading. And I mean, I guess it's true that it does depend on substrate abundance, but saying it only depends on substrate abundance is a bit of a stretch. So it's a bit of an over exaggeration. So I'd say C is the best answer there. Ah, yes. Now we get to talk about my favorite part of the exam. Yeah, well, guess what? I didn't get every one of them right. I messed this up really badly. Yeah, I messed this. It was a dumb mistake. So I knew what the answer was, and I misread the part, and I got it wrong. I think I got everything up here right though. Yeah, I got the translation stuff right. I had that stuff memorized. 15 though? Nuh-uh. Nuh-uh. 15, I messed this up real bad. I think when I did this, I remember this now. I think when I did do this, I did have the. This was a mistake. I shouldn't have done this. I had the R&R regulation memorized, so I didn't put it on my note card. And so, yeah, I knew it, but like I think if I had my note card, I would have double checked it. You know what I mean? And I would have gotten it right. But I didn't because I thought I was a smartass. My bad, video. My bad. Yeah, trying to overfarm the exam. I can't overfarm the. Yeah, we'll thank exam 2 for that. So I got taken back down to earth in 442, guys. Don't worry. But yeah, let's go ahead and just run through this. There's not much else to do. You'll notice that the grammar is not very good. In these. Yeah. In prokaryote translation, energy consumption occurs at multiple stages. During initiation, how many GTPs are used in initiation? 1. Where is the 1 GTP used? IF2, thank you. Nope, that is literally just an initiation. Yep. Hydrolyzed, which factor is it again? IF2, thank you. Yep. Yeah, I think you could say I think you could just say initiation factor and you get the full points, but I think he said I think he allowed
He lost 2, right?
it. Oh no, sorry, you're right. No, it says 0.5 awarded for just initiation factor. Never mind. Yeah, you're right. Yeah, I remember this. Yeah, I remember on the exam I actually raised my hand and asked him on the test. I was like, Professor Gu, do you want the actual number, or is it just chill to put the just initiation factor? He like, I want the number, and I'm like, okay, cool. I'm like, okay, dude, fine. So yeah, he wants the number. Please put the number. um, it's the GTPase.
like that's it. Like that's job GPA.
Yes, literally that's his job. Yes.
Right. IFGTPAs, like is that like that's it.
The IFGTPAs is IF2. Yes.
Oh.
Yeah. I can't remember what I put, but if you go back to my translation uh translation one video. If you go to my translation one video and like skip ahead, I literally labeled them for you.
Yeah, it's a 3, 1, that's 2.
Yeah, I think one sits below two if I'm not mistaken. So like a two attaches to one, and then three is the one that.
What's your video?
Yeah, so go just you don't have to rewatch it. Just literally just skip ahead and just find the section. I should have like put like in a pinned comment, but yeah, just skip ahead and it's all written down. So yeah. Factor to properly position the ribosome P site at the start codon. Start codon. That is the whole point of it. I think I had an aneurysm reading this part. I was trying to figure out what was going on. So it properly places the P site at the start codon, right? Because the methional tRNA has to bind. The mRNA prepared for elongation. In elongation, each amino acid requires how many GTPs? 2. Good job, guys. In total, in case you didn't know, it literally gives you step 1 and step 2. For one, so which one actually delivers the amino acyl tRNAs? EFTU. I genuinely think you guys have probably done these enough where you probably have them memorized. If you don't, it's fine. Just put on your note card. But I remember when I took this class, I had hammered these so many times that I had just gotten sick of it, and I just had it memorized. So EFTU. For translocation, it's EFG. Where is EFG used again?
I feel like move the
We're just running over from the 1 to.
I'm asking where it's used again. There's a second place where it's termination. It's used in termination again. Yeah. Also in my translation 1 video, so go look at that if you need to look at it again. Okay, in termination, how many GTPs are used?
1, 2, somebody 2, but like
2, but
but like what for like
I think for this one, you kind of have to read ahead for the rest of the quiz. So like it says, in termination, blank are hydrolyzed by blank factor. I think you need to kind of read ahead a little bit. So it's going to be 1. Yeah. 1 is hydrolyzed by release factor 3. Right? The dissociation of RF1/2. You have to put 1/2, by the way. You can't just put 1.
It's like, Tom?
I'm gonna be honest with you, I have no idea. I have no idea why it's called 1/2. I'm making the assumption that they are bound. Like it's some weird thing where it's like combined. It's in the lecture slide as well. It just says RF1/2. Genuinely, I don't know why it's done that. But I think you have to say like RF1/2, or you can say like RF1 and 2, which makes me think that they're not bound to each other. So I'm not too sure. I think it like I genuinely don't know. I don't know for that. But for the exam, you can just put RF1/2 and they can dock you points. If they ask you which one, they need to get a life. So
They tend to do that.
I highly doubt it for this. I'm gonna be honest with you. But you never know. I guess. I mean, they could decide to be mean, but yeah. So dissociation of that factors from the A site. Good. A site of the ribosome facilitating ribosome recycling. Additionally, ATP is consumed. At least 1 is required for amino acyl. Please write the whole thing. tRNA. Oh, that's a good question. Actually, let me see what the answer key says. I think you might be able to say just no. You have to say amino acyl tRNA synthetase. You have to say the whole thing.
Say ATR and say ATR.
Yeah. I think you can say AATRNA synthetase. That works too. But you have to say amino acyl tRNA synthetase because the enzyme is not just called tRNA synthetase. Because that tRNA synthetase sounds like just the thing that makes tRNA.
Well, I'm saying like synthetics. I don't know. This is country. So it's not say amino acyl tRNA.
Oh, I see what you're saying. Yes, you do because there's multiple of them. Yeah. It's required for amino acyl synthetase enzymes Yeah, it's weird because you just have to remember there are many different types of synthetases. Every amino acid has a separate one.
And would this whole class body isolate each other's theory?
Not yes, so they would all have different like tRNAs, but the synthetases there would only be 20 of them. There's only 20 amino acids, right?
That's-
Yes.
And those syntheses can also do the multiple TR.
Yes. Yes, because there's I think like not lysine, for example, has two things that code for it. I think it's AAA and like AGU or something. Um, the lysine synthetase has to recognize both of them,
Both types of
both types of tRNA. Yes, exactly. I think there's others. I think phenylalanine I think has four. There's a couple of them that have like four, right? So that kind of thing. All four of them has to be recognized by the synthetase. So
like some TRA work.
yes, yeah.
That's why
Yeah, yeah, IMP, yeah. So yeah, like with IMP, it's a bit weird. I can't remember I don't think they really test you on the IMP stuff that much. Not in the case of nucleotide binding. I think that has shown up on some of the past exams, though, right? It is the wobble, yeah, but wouldn't be too concerned about that. Enzymes charged to tRNA to each element, where ATP is hydrolyzed to. I got this part wrong. Actually, I remember this. It's hydrolyzed to AMP, if I'm not mistaken. I think I got that right. Yeah, it's hydrolyzed to AMP by consuming two of them. So when I say it consumes two phosphate bonds. The pyrophosphate is one of them, and then it cleaves the original one, right? So it cleaves the original bond, and then it cleaves the pyrophosphate bond. I got this wrong. I didn't get full points on this question because of this part. But yeah.
So the AMP two power. Yeah.
Yeah. So yeah, okay. So the activated intermediate involved in both de novo purine and pyrimidine, donor of ribose 5-phosphate PRPP. You don't have to write the full thing out, not triple P. During the committed step, de novo purine synthesis. In this, I believe it's glutamine. In this case, yeah, it's the glutamine. It's not the glutamine synthetase. It's the PRPP glutamine aminotransferase. Thank you. Yeah, this is the this is the um this is the main thing here. Also, like serves to donate the first piece of the purine ring. Hopefully, you remember it's a nitrogen that's attached on first. That's the other way of remembering it. But yeah, PRPP glutamine aminotransferase. I think is it aminotransferase? I can't remember.
yeah, I think so.
I'm trying to remember if it's aminotransferase or synthetase.
I think it's like
I think it's transferase. I think it's transferase. For the first step for going from yes, sorry, it's a mitotransferase. Yeah, it's a mitotransferase. Yeah, it's glutamine PRPP aminotransferase. That is the committed step. For purine synthesis.
Did this in example?
Yeah. So yeah, I was saying, did you do this in exam one? I guess it could show up again. You never know. Formation of the pyrinosome. This is a weird one. Pyrinosome, if I'm not mistaken. Yeah, this is one of those weird ones. Remember with the pyrinosome, right? It's usually very spread out, right? But then when you combine, when like the levels of when the levels are low, right, they start to like combine, right, to combine to make the whole complex, right, and so they start funneling into each other. And then blank serves as a common precursor during de novo purine. I'm pretty sure this is supposed to be IMP. Yep. So IMP serves as the common precursor, right, for guanosine and adenosine. The two molecules that combine during the rate-limiting step of de novo pyrimidine synthesis are oh yeah, I forgot you guys covered this. Yeah, did you guys cover this? Yeah, you did. Yeah, yeah. This is aspartate and carbamophosphate. Yeah, yeah. See, like this. Yeah, I need to do the video on this. I can't believe did I cover these? I don't remember if I covered these in my videos, so I might have to go back and look. The amino acid blank is used for purine glycine. Okay, if you see used in purine and not in pyrimidine, glycine, because glycine is used in the 3 backbone chain at the top, right? Both purine and pyrimidine rings contain nitrogen atoms derived from glutamine and this should be aspartate if I'm not mistaken. Yep, aspartate. Okay. Trying to remember where those are in the structures themselves. I can't remember off the top of my head anymore. It's been a while, but um, yeah, it should be aspartate for that. And then when DHATP is bound, the okay, so this is important. Inactive octamer, right? It has to be bound to the activity site if it's going to be the inactive octamer, right? Because if it bound to the specificity site, what is it going to make? UDP and CDP. Yep. If it binds to the activity site, right? Activity site? Sorry, no, if it binds to the specificity site. Already mixing them up. Yeah. Gosh, okay. The 4 molecules that can bind to the catalytic site. Catalytic site. Okay? Not the specificity site. I was being dumb. I put the specificity site stuff. Catalytic site. Okay? It's gonna be DUDP. I'm like trying to wing this off the top of my head. So wait, sorry. UDP, CDP, and it's GDP, and what's the last one? ADP? Yep, there you go. You see CDP, UDP, GDP, ADP. That's what you need to put there. I can't believe I missed so many points on that. I think it's like 4, yeah. Like I missed all of them. God, that's so frustrating. No, you're cooked. They're all wrong. Yeah. Purine nucleobases that are not salvaged or recycled are eventually converted into something, which leads to gout, uric acid. If you see gout, uric acid, right? I think you guys covered it a little bit, right? Oh, is it not going to be on it? Okay, I wouldn't worry about it then. Great. Okay. Forget about it then. Okay. For these, it's just enzyme names. This is just literally adding a Jesus. It's adding a methyl group, so it's just thymidylate synthase. This is RNR. This is NDK, because we're going from diphosphate to triphosphate. And this one is dumb. This one is phosphorybosyl transferase.
Question, why is it
Which one? Sorry for this one.
why isn't it HGPRT?
What was it? Sorry? HE.
HGPRT is one of like sure.
This is how
It's the body and the with PRP.
Oh, I'm like vaguely remembering this.
Yeah. So like, is the purity does this also ribotransferase maybe? Like,
I think it does. I'm just remembering what the HG stands for.
it's yeah, toxicity. I think it's just for my box type C.
Oh, is it hypoxyxanthine? Oh. Um, I think in this case,
It's funny, right?
it is a guanine, yeah. I think in this case, the reason I would go with phosphorybosyl transferase is just because you're adding this plus the phosphate. You're basically adding like
Isn't that PRP the one having to be catalyzed with it?
yeah, but the issue is like with PRP. Yeah, like I think you would catalyze it with PRPP. Yeah. Um. Yeah. It's a bit of a weird one. I'm not gonna lie. I'm trying to. I can't remember what I put for this. I'd have to take a look at the uridine slide stuff, not the uridine, but the uric acid slide stuff with the HGPRT. Um, but I would say like phosphoribosyl transferase. Whenever you're talking about like salvage pathway, generally speaking, and you're like you have just a base, right? And then you want to add the ribose back with the phosphates on there. Generally speaking, the best answer is always going to be the ribosyl transferases.
So the more general term?
to the salvage It's the exact same thing though.
Is it the same thing?
But it's just
Oh, is it just called HGPRT?
It's the specific one I think for quantum.
Yeah, here, I'll stare at it for a second.
Where is it? Somewhere. You.
then what took together.
I feel like you should be right to. This is a authorized it's a specific one. And have they should give you full credit for that. And put HGPRT.
This is just the PRT part of the.
Yeah,
It's just not
yeah, I feel like means like will be the policies will be question or anything. It's supposed to count as both. Like, yeah, even if they didn't give you points for it, you are sure.
But yeah, I would totally argue that if they didn't give it to you. That seems a bit extreme to me if they don't give it to you.
super.
Yeah. So yeah. Just generally, if you want to be like super safe, I guess you can put like just phosphoribosyltransferase transferase if you want, but I feel like that's totally valid. So yeah, yeah.
So you curious how the EF2, EF1, EF3 initiation factors?
I believe so. Yeah, we just call them EIF2. We just say E in front. So EIF2, EIF3, EIF1. Yeah, I have never heard of a case where they're not included.
Like the useful, I feel that there was like a methotrexate.
Oh, I didn't even see this.
Oh,
I don't know if you've ever learned that. Methotrexate is a potent anti-cancer. I mean, I know the answer. It's gonna be THF reductase, I think, or it's gonna be yeah dihydrofolate reductase. I don't think you guys talked about that. Yeah.
Why was that?
I think the main thing that you can get from this is like even if you don't know what this is, potent anti-cancer drug that starves the cells of this. It specifically starves them of the T thing, right? So based off of that, through inhibition of this enzyme, right? And you do have to know this, the dihydrofolate reductase.
I DTMP synthetase, that's direct. Like it says, like if I did, I would say just inhibits the actual enzyme that just alloys.
For the thing, yeah.
Cause like there's like 3 different enzymes before like that.
Yeah, I'm trying to see if there's like a. Is there a reason that you couldn't do that?
Like there were thinking about this example too was like why they why would it be more than TMP?
Yeah, yeah. Yeah, I mean, like, I'll talk about that if I need to. Yeah, I can come back to this if we need to, but I'm gonna try and finish this if I can. I don't think I can, but I'm gonna try. GGC. So for this one, right, you identify the gene sequence using a codon table, right? You need to do the whole thing here because it tells you that this whole thing is a C-terminal sequence. So the whole thing needs to be translated, not just the end part of it. Right, because it tells you that you need to do the whole thing. So determine the C-terminal amino acid sequence from this enzyme, and it tells you, yeah. So it ends with this. So the whole thing is the C-terminal thing. So you can just like do the thing for that if you want. I'm not going to do the whole thing, but it's going to be glycine, phenyl, and glutamine, and then you're done. If the GGC codon is mutated to a rare thing, how would it affect translation efficiency? It would decrease because if it's a rare codon, there's not going to be any like of that tRNA present really. So tRNA present, so it slows down translation, something along those lines, something like that. You isolated the mRNA of this enzyme with a short poly A tail and use it to do that. Provide 2 examples of single base mutations. This question I got stuck on when I took the actual test. So if you have this, right, remember, mRNA with a short poly A tail. That's very important here. Poly A tail is very important. Okay, and you want it to end with a C-terminal lysine. Okay, so what we can do is the first thing we can do is CAG. We can mutate that to AAG. So AAG, right? So here's the thing: we have triple A is lysine and AAG is lysine. So those are the two things we have. If we change the C to an A, so CAG, CAG, AAG, point mutation, exactly, right? It's a missense mutation. So that's one option that works, right? The other option that works is I destroy the stop codon.
Oh yeah.
If I destroy the stop codon, so I don't know, just UGA to, I don't know, UCA, I don't know, whatever. Right? This would result in the final thing being codon being triple A, which is also lysine. Right? All I'm doing is, so normally, right, the stop codon stops the thing from translating afterwards, right? Does that make sense? If I remove the stop codon, it just keeps going. Yeah, it just keeps going to the end. Yeah. I'm telling you, when I took this exam, I got stuck in that question I asked the TA and I was like, there's no way to solve this. And they were just like, look at the question carefully. And I was like, yes, yeah, I know. I know. That's what I was thinking too. But you have to think about the whole sequence. Everything that's there. So the poly A tail is there as well. So please, yeah, be very careful with those kinds of things. Okay, for C, you add the above unmutated mRNA to two cell-free translation things, one with a release factor, one without. Okay, describe the products corresponding to the observed bands with and without the release factor. So if I have with the release factor, right, it's just going to be the full peptide chain, right? It's going to travels, I don't know, far in the band, right? Something like that. Far in the gel, sorry, in the gel. If I don't have the release factor though, right, the tRNA never gets released from the peptide chain, right? So it's going to be a tRNA plus peptide chain thing. So it's going to be tRNA plus peptide chain complex, which travels not as far compared to just the peptide because it's heavier, right? It's heavier, so it travels less in the gel. Does that make sense? Right?
But heavier is heavier is more of a fair.
Yes, higher up. Yep. Because you started from the top. Yes, less far. Did I say not as far? Okay, yeah. So it wouldn't travel as far, right? Because it's heavier, right? Does that make sense? Yeah. Okay. Yeah, I'll come back to it afterwards. I just want to make sure I get through the test. If I need to. Explain how the process, presence, or absence of release factors can result in different patterns. For this, I believe, yeah, I think like the presence, when it's just asking for the mechanism, right? So when the RF is present, we can hydrolyze the peptide from the tRNA, right? When it's not, we can't hydrolyze. That's it. That's all. Yep. Yep. And that's all they want, really, if I'm not mistaken. Peptide transfer is presence of release factor. Hydrolyze in the presence of release factor. Yeah, they wanted you to describe it in both cases, but like you would say like in the presence of release factor it was not it was hydrolyzed, and without the release factor it wasn't hydrolyzed. They just want you to say both, so you can just like copy paste this and then just like do the same thing, but say it when there isn't a release factor. You know what I mean? Yeah, I actually put that on my test and I got full points, so you can do that. Yes. Yes. Yep. That is technically, it's an IMF. It's an intermolecular force, so they will fall off because SDS page disrupts them. It won't disrupt the tRNA though because it's a covalent bond. Yeah. So, yeah. It's just yeah. For this one, provide an explanation. Okay, for this one, I'm not going to bother going through the whole thing, but you design a hyperactive thing, hyperactive mutant of thymidylate synthase with increased activity compared to control cells. Provide an explanation, and then you observe that it has a decreased activity in RNR. This is literally just going back to this. and saying, okay, if I have high levels of DTTP, right, I'm literally just doing a flow diagram. I'm going from high levels of DTTP, right? I start here, goes to this, right? Goes back to this, goes here, makes this, and then turns it off. You have to write that all out in words, basically, right? Just draw a flow diagram, if you know what I mean, like that kind of thing.
Wow.
Pretty much, yes.
That's the PvP over
Yes. Yep.
That would have been
Not that.
PvP and then that would a lot of PvP.
Yep.
PvP and then that would go.
And then when targeting rapidly dividing cancer cells, use chemotherapy drugs trying to minimize the effect on normal cells. Why would thymidylate synthase serve as a better target than dihydrofolate reductase? There you go. That's that question there. So why would thymidylate synthase be better in this case? It would just be better because cancer cells, they divide a lot more, right? If you mess up a dihydrofolate reductase, you also mess up a lot of the amino acid pathways and a bunch of other pathways that are really important for normal cells. But thymidylate synthase is very specific to dividing cells. And cancer cells, they divide a ton, so it's a better option to inhibit that instead. Yeah. It can be.